Hi, this is Miss Slitton, and we don't know where everybody else is, but this is AP Unit 3 Essay Review. Say hi.
Hi.
Right. So you have several questions. One on there, this is small for those of you at home. It says all new cells come from previously existing cells. We know that from the cell theory. The process of cell division, which involves both replication of the cell's nucleus, and right now in chapter 12 we're learning how that process goes, right? DNA unwinds, unzips. What enzyme? DNA. Helicase. All right. Replication of the cell's nucleus and division of the cytoplasm. Now, replication and division of the nucleus, when we talk about division of the nucleus, what stage is that called? Mitosis. And division of the cytoplasm is? Cytokinesis. There are two types of nuclear division, mitosis and meiosis. Name and describe each stage of mitosis and then meiosis. And please describe the importance or role of meiosis in gamete formation and variation in sexual reproduction. So there's a lot in that, right? So first of all, you would, in a situation like this, sometimes I'll say, for instance, I might say 2n equals 2. Draw for me, and I'll name a stage. What would prophase look like? What would metaphase look like? What would anaphase, telophase look like? What would prophase 2, metaphase 2, anaphase 2? Do you see in through all of those situations? So could you do that? Okay, so tell me, what would metaphase look like if that were true? What would metaphase look like?
So
if 2n equals 2.
Then you have one homologous pair.
I have one homologous pair, but what would metaphase look like?
So you just have one in the middle like that.
One here, and that's it? And one underneath, exactly. Like this, right?
Wait, isn't it horizontal? Or?
No, okay, if you drew it like this, what stage would that be?
No, I mean metaphase 1.
Right, right. So we want it to be lined up,
Oh, wait.
right? Okay, then what would metaphase, I forgot some letters in there. What would anaphase look like?
You would have the one sister chromatin going to each side.
Yep,
of the spindles.
they'd get pulled like this, right? And you would have microtubules, right? Yes?
Mm-hmm.
And then telophase 2 cytokinesis would be something like, oh, I changed the color. I'm sorry, but you know what I mean.
Yeah.
Right? Okay, that would be different than, what if I said instead metaphase 1? What would that look like?
You'd have the homologous pairs next to each other.
Homologous pairs next to each other like this. And then anaphase 1, you would see what?
They would go separate.
Yeah, they would go into separate cells, right? And then this would be what metaphase 2 looks like, and then anaphase 2,
So you have just one.
right? Yes?
Would we have to draw like the mitotic spindle?
I would put like a few on there, but you know, yeah. Okay, so we would be able to name and describe each of the stages, and then the next part says describe the importance and role of meiosis. Whoa. Okay then. Okay. Okay. There we go. Name and describe the importance role of meiosis in gamete formation and variation in sexual reproduction. So just referring to gamete formation, what are the two ways we can get variation?
Oh, independent
Assortment and
Yes. Let's do them in order of how they would happen.
so crossing over would occur.
And what stage does that happen?
In prophase 1.
Prophase 1, we can get crossing over, right? And would you be able to give me a definition of crossing over?
Yeah.
the shuffling.
It's in the homologous pairs, the non-sister chromatids cross over their alleles.
Exchange.
Exchange.
Yes, they exchange segments of the chromatid, right? Okay. And then where would be the second place?
Metaphase.
1.
And what happens there?
Independent.
Independent assortment, right? And so each homologous pair can assort independently of all the other homologous pairs. You can get any combination of alleles. Where do you not find that? If the genes are linked, right? You're not going to get independent assortment.
Wait, I don't get what linked genes are.
Linked genes are when, if I talk to you, let me tell you what linked genes aren't, and then we can kind of see it here, okay? if I said there is chromosome A, right, and both of these would have big A because they're sister chromatids, and I'm just, doesn't have to be this way, but say this is little a, and say down here I have R's, right? So if they lined up like this, I could get big A with big R and little a with little r. That's standard independent assortment, right? But if I had another pair of alleles on here, B, right? I'm going to tend to get big A. What are my options I'm going to tend to get? I'm going to tend to get big A, big B, and big R. Or if this is flipped the other way, I'll have big A, big B, and little r in the gametes.
Oh, okay.
See how these are traveling together?
Mm-hmm.
Oh.
Those are right here. These are linked genes.
Okay.
But if
So they don't do independent assortment. The only thing that breaks up independent assortment, the only thing that says no to those linked genes is crossing over. Crossing over, if this flips right here, well, I'm sorry, if this flips right here, if these two exchange, then I can end up getting a little b here and a big B here. So now each one of these chromatids is very unique. I have a big A, little b, big A, big B, little a, big B, little a, little b. That's the only thing that's going to break up those linked genes. Does that make sense? Okay, so that's in gamete formation, and then in sexual reproduction, which would be a separate thing. This is just in meiosis where I get variation. But the third way I can get variation as a result of this process is through what?
fertilization.
Yeah, random fertilization. But just make sure to keep those in your mind, right? These are a result of the process of meiosis. This is a result of sexual reproduction. Okay. All right, so that is that essay prep. And then let's look at the next one. Cancerous failure of genetic control. What are the general characteristics of a cancer cell? So what I would go do is look in your notes and look at, you know, what we listed as typical of cancer cells. You know, that they have very little cytoplasm, abnormal nuclei, all the things that we listed that were typical of cancer cells, physical attributes that you could see. Okay? B, what two types of genes in particular malfunction in cancer cells, and what do these genes do in normal cells? So what are the two genes we talked about that we need to deal with?
The tumor suppressor.
What? Tumor suppressor, and what else?
Proto-onco.
Yep. So the normal ones are proto-oncogenes and tumor suppressor genes, right? If proto-oncogenes, what they normally do is they stimulate the cell cycle. They say, go cell cycle, and as long as they're working functionally, we're fine with that, right? And normally tumor suppressor cells, they'll stop the cell cycle and stop the cell cycle, they'll check to see if everything's going well, and if it's not going well, then they'll kill off that cell, right? Apoptosis.
The gas and then the brain.
Yeah, so, yes. So this would be like the gas, and what gene do we know? What's an example of a proto-oncogene? RAS is like stepping on the gas. So that's an example, but it is a proto-oncogene. If it goes bad, okay, so there's normal RAS and it's working fine, turning on a cell to make a cell make more cells. You know, that's fine. If it goes bad, we say when a good cell goes bad, it's become an oncogene, because now it's not in control. Now, still, we might be all right as long as our tumor suppressor cell is there to wipe out that, then we'll be okay. But if we have a bad RAS, and on top of that, now our tumor suppressor gene has undergone some sort of mutation, now we have no way to stop it and kill it if it's not fixed. And an example of one of those is the what?
P53.
P53 gene helps code for P53 proteins to stop a cell that's out of control. So these are examples of this. This is an example of this. Okay? All right. State each of the conclusions reached by Mendel in his work on the inheritance of characteristics. Explain how each of the following deviates from these conclusions. So first, tell me the three conclusions he had. Law of dominance.
Law of segregation. Law of independence.
Okay, so law of dominance is saying that you can mask a recessive gene, right, with a dominant gene. Like for pea plants, tall is dominant over little t, right? And then law of segregation says that the T's will segregate from each other when you form gametes. So you only get one T. If these are going to go be sperm, okay, they only get one T in their sperm. That's the law of segregation. Because this happens in both anaphase 1, you separate homologous pairs, and anaphase 2, you separate sister chromatids. So you're only going to get one copy of that. That's what the law of segregation says. The law of independent assortment says you could get a big T with a big R or a big T with a little r, right? That's our law of independent assortment. We just talked about that, right, during metaphase 1 when they line up. Those are the three laws. Then we want to explain how each of the following deviates from that. Well, autosomal linkage, that's a normal thing. That's not like, oh my gosh, we've got trouble here, right? Autosomal linkage says if you have a chromosome and if T and R on that same one, they're not going to do independent assortment because T is going to go wherever R goes, right? So any gene linkage would then negate the law of independent assortment. Okay? Sex-linked inheritance, also normal. But in sex-linked inheritance for women, right? For women, dominant over recessive still works. But it doesn't work for males, right? Because in order for a woman to get a sex-linked disease, she has to get 2 of those, right? So that means both her mother and her father have to be able to contribute to that. But that's not, law of dominance doesn't work for males because they will only get one type of X. So you don't look at normal dominance and recessive. You know if it's going to be a boy, dad's given the Y. So mom will give either one of hers. So you don't have dominance and recessive on the X chromosome. You do on all their autosomes, but not on their sex chromosomes. They get what they get. They get 1 allele, 1 shot. As women, we get 2 alleles, 2 shots on everything. Guys have it on everything except their sex chromosomes. So the law of dominance does not come into play with sex-linked traits. Yes?
To have an X-linked trait for the inheritance, it has to be a recessive one, right?
No, they're dominant ones too. I'm just not giving you any.
Okay.
The reason is they'll start to become hard. It's a real subtle to see a sex-linked dominant and an autosomal dominant, and I'm not holding you responsible for that. But there are sex-linked dominant diseases.
Okay.
Yeah. Okay, and then polygenic. Okay, so on all of these, we're saying one set of allele codes for that one trait that we have, right? But polygenic is not straight up dominance, because in a polygenic trait, poly means what? Many. So like for instance, for our skin color, right, there could be a combination of A's and B's and C's. It's polygenic to give us our skin color. So it's not like there's dominant or recessive. You know, it's not straight up dominance. It's a combination. It's an amalgamation in order to create that trait. Good? Okay. So put another one on there. Discuss the variety of gene interactions listed below. Be sure to explain the inheritance pattern as well as give an authentic example of each. So incomplete or partial dominance. We talked about that. There are 2 simple examples you could do for that one.
The red flower and the white flower.
Red flower, white flower makes pink flower. Straight hair, curly hair makes wavy hair. So on incomplete or partial dominance, it does not follow the law of dominance. You don't have one allele dominant over the other. You get a little bit of each of those getting expressed. So it's not completely dominant. It's really, incomplete partial dominance is really usually just basically co-dominance, but the way it expresses itself, it looks blended. But it's like the red flower is dominant and the white flower color is dominant, like blood types. It's just you're laying down a layer of red pigment and a layer of white pigment, so it appears to us as pink. It really is just a part of codominance, but when it looks blended, they call it partial or incomplete. Okay, epistasis. what was the one for epistasis that we looked at? Epistasis means what? One pair of alleles, right? In particular, one gene codes the fate, controls the fate of another, the way another pair expresses itself. So we looked at that for like coloring, like in dogs or rats, that you have to have, or you could do it in guinea pigs, where you have to have at least one color gene. And then from that, you could be big B, like a black guinea pig, big B, little b, still a black guinea pig, little b, you're going to be a brown guinea pig. But if you have instead two little c's, no, no, and no, you're just going to be an albino guinea pig. It doesn't matter what these alleles say, because if you have two little c's, then you're going to be albino, because it's this right here that says lay down pigment. It tells you to put pigment down. This just tells you what kind of pigment you will lay down, right? If you don't have the lay down pigment gene, you're not, doesn't matter if you said brown or black. Doesn't come into play. All right. What was pleiotropy about? Remember that?
It's like a single, when a mutant gene creates multiple traits that are like distinct.
Yeah, so you have a whole syndrome that's associated. And we talked about sickle cell disease as an example. We talked about Down syndrome. Anytime you have a syndrome, okay, it means, like, for instance, if that one set of genes, right, codes for many different traits out there. One pair of genes coding for many different traits that are all grouped together. And you could use sickle cell disease as a good example of that. Because the reason why I say Down syndrome is really you have an extra copy of that, and that's why there's a problem. So I gave you a slide on that on sickle cell. Okay, multiple genes are polygenic inheritance. What are some polygenic traits that we looked at?
Blood type.
What?
Blood type.
No, that's multiple alleles. That's
okay.
E. We already did one already. Skin color was polygenic. I told you intelligence was polygenic, right? Where multiple genes are coding for one single trait. C and D are kind of opposite of each other. Do you see that? C and D are opposite. C says I have one pair of alleles that codes, right, for a whole bunch of different pleiotropy. This one pair of alleles has a bunch of different traits, whereas multiple gene says I have several different alleles coding for a single trait. Do you see the difference? Yeah, so pleiotropy, one pair of alleles, a whole bunch of traits, multiple or polygenic inheritance, multiple alleles coding for a single trait like intelligence, etc. All right, and then multiple alleles, your best example there is blood type. So you don't just have big I and little I, you have big I A, you have big I B, and you have little I, and so you can have a combination of any of those to get your final blood type. All right, I'm going to keep going. Experiments by the following scientists provided critical information concerning DNA, briefly describe each classical experiment, and indicate how it provided evidence for the chemical nature of the gene. Do you remember Hershey and Chase? What did they work with? Bacteriophage.
Oh yeah.
Right? And so that was this guy, right, on the bacteria, and they had P32 and S35, right? And what were they looking for there? What was the transforming agent that the virus was giving to the bacterium to make it make more viruses?
The DNA.
Yeah, the DNA, and that was indicated by the radioactive radioactive phosphorus being found inside the DNA. Okay, Griffith, what are we thinking about there?
Rats.
Yeah, those rats or mice, and Avery, McLeod, and McCarty, and what role they played. We talked about that today or last class, depending what period you're in. And remember, they were working with, you want to remember that they were working with bacteria, right? And one was a smooth bacteria, one was labeled the R, the rough, And you want to just make sure you know that heat killed S, which was normally lethal, was mixed with live R, and the R got transformed into S. And then Avery, McLeod, and McCarty are the ones who did the experiments to show that that transforming agent was DNA, because they used enzymes, degradative enzymes. And when they broke down the DNA, no transformation. When they broke down the proteins and the RNA, there was transformation. Misselson and Stahl, I just mentioned both classes. Misselson and Stahl, they did the experiment to support semi-conservative replication. Just write that down as a note. There's more to that experiment, but it's not one of the ones that I believe the new test will have you memorize. But it is in here. But basically, their experiment supports semi-conservative replication. And I'll talk about that to the whole class. Okay, on this one, describe the biochemical composition, structure, and replication of DNA and any enzymes that are required. That we just learned today, right? So when you talk about the composition of DNA, you want to make sure you understand that it's made out of base, sugar, phosphate, and that is a nucleotide. You'd want to name the 4 bases, right? You want to tell me about deoxyribose. As far as the composition, you have deoxyribose, which is the sugar. You have nitrogenous bases. And you have adenine, thymine, guanine, and cytosine. You'd want to tell me of those who are the purines and who are the pyrimidines. And you'd want to talk about what that means. Okay, and then you have phosphate groups. Okay, that's the biochemical composition. The structure is, you're going to tell me what for the structure? Yeah, I would say 2 strands of nucleotides, right? Held together by what? H bonds, right? I would say A, 2 H bonds to T, and G, 2 H bonds to C. I'd talk about the phosphate sugar backbone, and I'd be able to do like some simple diagram of it.
Should we say it's anti-parallel?
Yes, anti-parallel. Yes, I would say that's great.
And
Definitely.
The 5' to 3'.
5 prime, 3 prime. Yeah, just do a simple, a simple picture to show that you know how it's set up. Okay. Then replication, right? What are the names of the enzymes you're going to tell me about, or structures? Helicase. And I would probably do helicase topoisomerase. What else?
Polymerase 3.
Remember SSB holds the 2 strands apart. Okay. Then what would you do?
Or the primase.
Yes. Make sure you know RNA primase.
Oh, okay.
Okay. And it lays down 10 base pairs of RNA nucleotides, which is called the primer, right? So make sure you tell me all of those things. And then I would say you need to do it 1 time on the what strand?
Leading.
And multiple times on the lagging strand. Okay, what's the next one you would use?
Polymerase 3.
Yep, DNA poly 3 works off primer, reading, yep, 3 to 5, making what?
5 to 3.
5 to 3. Good. Who's next?
DNA polymerase 1.
Yep. Okay, and DNA polymerase 1 removes the primer and replaces it with nucleotides. What kind of nucleotides? DNA. It removes the RNA nucleotides of the primer and replaces it with DNA nucleotides. Yes?
So do both polymerases, do they both pick off the primer?
No. DNA polymerase 3 only works off of the primer. DNA polymerase 1, he is the one to save you. He will remove the primer and replace it with the correct.
Okay.
Yeah.
Okay. Got it.
And proofread along the way. It's like he's proofreading. No RNA here. Yeah. No. Okay, and then it'll replace. Okay, and then who's next? Ligase. Now, you have the strands held together here by the hydrogen bonds, right? But you need to seal the phosphates and sugars wherever you took primer out and replaced it with DNA. Now you've got to get the phosphate sugar backbone sealed together, and that's what the ligase is doing. Okay, because DNA polymerase 3, when it works off of it, it's doing the phosphate sugar backbone, right? But when the primer got taken out and put and replaced, you need to seal it up with all the Okazaki fragments need to get all connected together.
Okay.
Yeah. And on this one, when we talked about the DNA polymerase 3, that would be a good time to talk about the Okazaki fragments during the DNA polymerase 3 time. Okay, that's that. Are you glad you came today?
Yes.
Okay, next one, describe the steps of protein synthesis. We can't do this yet because we haven't learned this one. So we'll save this one for
Part 2.
Yeah, part 2 review. What?
When is part 2?
We'll probably I'll just say it, look at this question when I teach it to you and go, hey, here's that question. We've done all the other ones, yeah. All right, this was the grid-ins. I cut and pasted the grid-ins. These are probably pretty easy for you, but let's just double check them, okay? Yeast cell in the early portion of interphase of meiotic division has 24 FGs of DNA. If a yeast cell completes meiotic division to form 4 haploid cells, how many FGs of DNA would be expected in each haploid cell? Now, I don't know what's coming in your mind right now, but you want to go logic first. What could you tell me about a cell that is undergoing meiosis? You go from being what to what?
A diploid
To haploid. Now you know it's still going to replicate its what? DNA. And it does that, and the first division separates the homologous pairs. Second, and then it separates the sister chromatids. So in your mind, are you coming up with a number for how many FGs we would expect?
Yeah. We expect 12.
12.
12. Okay, 12 each, but like it says 4 cells.
Yes, but you got to keep in mind, if I tell you the 2N cell equals 24 FGs,
Okay.
then what's the N cell going to be, right?
12.
It's got to be 12.
I thought it said in like all the haploids.
But do you see what I'm saying? You've got to think, do you need me?
I do, but it's a tech thing, and now it's just a question. I'll ask you later.
Okay, no problem. You just got to think big picture so you don't make that mistake, okay? And because these are the kind of questions, these grid-ins, where you can like read too much into it and make it more difficult than it is. So you go, okay, I know in meiosis I go from diploid to haploid, so if I start out with 24, I've got to have half as much, right? Because the 2 gametes have to get together to form a diploid zygote, right? So all the chromosomes replicated, and then you only sent 1 chromosome 1 and 1 chromosome 2. So you're going to get down to half the number, okay? And then number 8 on your grid-ins, in a pea plant, T is tall, little t is dwarf, R is round, and little r is wrinkled seed. If a TTRR plant is crossed with a TTRR plant, what percentage of the offspring would be tall and wrinkled? How could I write tall and wrinkled here? Tall.
Tall is T.
Big T, anything will be tall, right?
Yeah.
How will I be wrinkled?
It will be R.
Tell me what kind of R?
Little r,
Little r, little r. So I'm wondering how many will turn out like this, right?
Mm-hmm.
So what I need to do in that situation is I need to cross just my T's, right? Right? So how many of just my T's will turn out big T anything?
3/4.
3/4, right? And then very carefully cross your R's, right?
Very carefully. I'm very careful.
I'm very careful.
Oh yeah.
How many of my R's will turn out little r, little r?
1/2.
1/2.
So do you multiply
So,
those?
yeah, 1 times 3 is 3. 2 times 4 is 8, 3/8. Yes?
How did you know to multiply those and not add those?
Okay, because I want to say whenever you're looking at, I need the T-ness to come out this way and the R-ness to come out this way, I do what's the probability of the T turning out times the R. There's not another way to get it. What we did right here is we added, this was your added part. Because we said big T, big T, big T, little t, and big T, little t will get us T something. That part we added, 1/4, 1/4, because there was 3 different ways to get it. So that was add, add, add. When I say I want this state of T-ness with this state of R-ness, I multiply the probabilities together.
Okay.
Independent events happening at the same time.
Okay.
If there's more than 2, would you still multiply them all together?
What do you mean? Tell me again.
Like if it's more than 2, like say it's like multiple letters, like 3 letters.
Okay, so let's say the parent was this. Okay, and let's say I wanted to know how many would turn out this and this. Then what would it be? It would be still 3/4 times 1/2. Can you see that cross in your head? Yeah, 1/4.
Okay, you multiply them all together.
1 times 3 is, yeah, because I'm saying I want all 3 of these events to happen at the same time. I have 3 ladies going into labor right now in the hospital. What are the odds that this, and this all have a girl at the same time? Well, it's a half a chance here that she'll have a girl, half a chance that she'll have a girl, and half chance that she'll have a girl, half times a half times a half. There you go. All at the same time in that order.
In that order.
Yeah. Okay, we good on those?
Yeah.
Okay, so then, where are we? Here.
There was one
Here.
That one.
And this is the one I worked out for you, and it's all in your group shared notes, but just so you know, so we can see here, when we cross gray body, normal wings, if we were going to do, you know, if we thought we were crossing this and normal wings, we would have thought, I bet 9 are going to be gray and normal, 3 are going to be gray and vestigial, 3 are going to be black and normal, and only 1 will be black and vestigial. But it didn't turn out that way. I didn't get that independent assortment. The bulk of them turned out the 2 dominant, gray and normal, and the 2, thank you, sweetheart, and the 2 recessive traits, black and vestigial. And only a few, when you see only a few having the combination, you know, it's not like even Steven combination, and then you're like, something's up. So these were a result of crossing over. So when you want to know map units, you just take up how many are the ones that were a result of crossing over. Well, that would be 103 plus 92. I don't know what I'm writing. Plus 92 over the total amount of offspring. I'm just the percentage of crossing over. So that would be 482 plus 472 plus 103 plus 92. And you're just getting a percentage that are crossing over. Oh, and that's all of that. And what's this? Okay, let me just show you this one, because it'll be easy real quick, okay? So this one is saying, below is amino acid sequence for 6 species critical to whatever gene. Each letter represents a different amino acid. Amino acids make up proteins, right? So assuming mutation rates are equal for all the species over time, based on this data, which letter? So they only want us to look at B, C, D, and F. Those are the only ones you have to look at, okay? Which ones represent species least related? Okay, so just compare A with B. And how many differences do we have between A and B?
Just one.
A, T, E, T, I, P, K, S, S, right here?
Yeah.
And then T, S, S, that matches, right? Okay, now let's do A and C.
That's all the same.
Exactly the same?
Yeah.
Okay, now let's do A and D.
Oh, there's one difference.
There's an N.
And an N. An N.
Right out here?
And there's an N as well.
And an N. Oh, yep, right there. You see it? Okay, now let's do A and, because it didn't ask me to look at E. A and F.
V.
Is that it?
Yeah.
Okay, so how many differences between A and B? 1. How many differences between A and C? 0. A and D? 2. How many differences between A and F? 1. Who had the most differences? D. D is the least related.
Oh, wow.
See? Easy peasy, lemon squeezy.
So it's just like discrepancies.
Yes! Okay, so we've reviewed all of the essay questions, all of the gradients except for 1.
Thank you.
Good job. Super proud of you.
Thank you.
And we did it in 35 minutes. Have a piece of toast.